Hello, everyone. Thank you for joining us today for the revision lecture. So this is part one of the 2 lectures that we are delivering as a part of this series. And today we'll be covering Unit 3 of VCE Biology. So firstly, I would introduce myself. So I'm Navneet. I'm the VCE Science Lead at TFI. And accompanying me today is Eve Chen, who's the VCE Science Tutor. And we also have Joey, who is the VCE Team Lead. So before we get into the content, just wanted to share a few other platforms you can log into. So here's the email address if you have any queries. We also have an Instagram and a Facebook page. You can also check out more information about other lecture series that we are delivering on our website as well. But in terms of the content itself, should there be any questions, feel free to put them in the chat. Just to continue the flow, we won't be able to answer them during the lecture itself, but there will definitely be opportunity to ask questions at the end. Or this meeting is actually getting recorded, but if you are comfortable in doing so, you are more than welcome to unmute yourself at the end of the lesson and ask a question if you have. Meanwhile, let's get started. So first topic for the day, plasma membrane. Just a reminder, plasma membrane is a phospholipid bilayer, i.e. it is created via 2 layers of phospholipids, which contain a hydrophilic head and a hydrophobic tail. So hydrophilic means water-loving, which is why it's on the extracellular and intracellular environments. And sandwiched in between are the hydrophobic tails. This layer overall looks somewhat like this on the cell membrane, which is why it's also referred to as the fluid mosaic, because as you can tell, due to the different components that we have in here, it looks like a mosaic wherein different units have been joined together, and it is quite fluid. So each sort of phospholipid pair has some flexibility of moving around. In terms of the fluid mosaic model, the main elements you need to know about are the proteins. So there's 2 main types of proteins involved there. Some are transmembrane, where they span the whole membrane, and some are just peripheral, where they're only on one end of the membrane. In terms of the structure, those are the important parts you need to know. But in terms of the main purpose of plasma membrane, which is regulating movement, there's 3 main methods of moving. So there's simple diffusion, which is anything that is small enough and does not possess a charge. Sort of things that are like, I'm not going to be in anyone's way, just let me swim through, can just go based on their concentration gradient. Things that need a channel, maybe because they are charged or because they are a bit too heavy for the pores, that actually gets sent through specific gateways. And then there's things going against the concentration gradient, which is called active transport. So in this one, you obviously do need a channel as well, but the term active comes from the fact that it needs ATP as well, because you're forcing things against the concentration gradient. So that's how little molecules can go across. But when we talk about bulk transport, in this one, we have 2 main types. So we have exocytosis and endocytosis. In exocytosis, we're talking about molecules leaving the cell, and in endo, it's about absorption. But when it comes to proteins leaving the cells, they have to follow this path. So it'll start with the rough ER, which is the endoplasmic reticulum. And when we say rough ER, rough refers to the fact that it has ribosomes attached to it. So from the rough ER, it goes into the lumen of the endoplasmic reticulum. From there, it can go into the sorry, I'll just continue as we were. Welcome again, but I'm just going to continue as we were. As I was saying, we have rough ER, then it goes to the smooth ER, and then we have Golgi apparatus. So Golgi apparatus is, if we remember, just a structure looking like squiggly lines. There's a cis phase and a trans phase. Eventually, the molecules enter from here, come out from here as teeny tiny vesicles, and those vesicles will then take things out of the cell. When it comes to vesicles, there's transport vesicles that take proteins from one organelle to another, but there's also secretory vesicles. So it's important to know the difference, because one type helps them from going from one organelle to another, and the other type takes them out of the cell via exocytosis, which takes us to the next topic. Exocytosis, like I said. So essentially, things will be packed in a vesicle. The vesicle fuses with the cell membrane temporarily, and things will be expelled out of the cell. When we are talking about things coming into the cell, it's called endocytosis. Again, another type of bulk transport. Same sort of concept applies. Vesicle fuses with the cell, goes in to another vesicle, and then goes to the designated area, of course. But there's two types of endocytosis. One is called phagocytosis, which you guys will come back to later on in immunology as well. Yeah, immunology, my favorite topic in biology. But anyhow, the other type is pinocytosis. Pino, whatever you prefer, essentially when things are liquid. So as you may remember in M, when we discuss about how pathogens are engulfed, we usually refer to it as phagocytosis because that is a solid being consumed. If it were a liquid, it would be called pinocytosis. So, okay, let's actually have a go at this question. So I'll give you maybe a minute or so to have a read through this question before I give the answer. Meanwhile, feel free to chuck your answers in the chat if you're comfortable. Okay, so we do have an answer in the chat. So the answer was B, which is actually the right answer. So just to go through the other options, the reason why it was B is because B would be, sorry, so B is small enough and it does not have any charge that would interfere with the structuring of the protein, the plasma membrane, which is why it does not need a protein channel and can just swim through based on its concentration gradient. Sodium ions, however, as the word suggests, ions are charged, which is why they need additional support. And polar protein molecules, again, the charge, ribonucleic acid, same thing. But oxygen is good enough to just go across. So moving on to the next topic, which is nucleic acids and proteins. And as always, if there's any questions, feel free to put them in the chat and we'll come back to those at the end. In nucleic acids, the main thing you need to know is the monomeric units are nucleotides. The nucleotide is made up of a phosphate head, a deoxyribose sugar when it's DNA, and a nitrogenous base. The nitrogenous bases can be of 4 different types for DNA and 5 in total. So when it comes to DNA, it's GCAT. My biology teacher used to always say good couples always together. So good as in G will always pair with C. A will always go with T. When we talk about RNA, however, that's when it'll base pair with uracil instead of T. Speaking of RNAs, there's 3 main types of RNAs: messenger, tRNA, and rRNA. We'll meet these again in translation, but this is just a slide for you to refer back to if you need a summary on these 3. Moving on to proteins. So from the DNA, we eventually have to go to proteins, which is just a polymeric unit of amino acids. This is just highlighting how a typical amino acid looks like. And just a reminder that the entire set of proteins in an organism is referred to as proteomes, and the study of these proteins is called proteomics. Having been through the whole course, you would obviously have a somewhat understanding of what enzymes are and how there's peptide hormones. We'll come back to those as well, but this is just to reiterate that proteins can be in several forms. Moving on to the structures of proteins. So there's 4 main structures. Primary refers to the main chain of amino acids. Secondary is the folding into alpha helices, beta sheets, or there might be a third type as well, which is called random coils. So that's when things cannot be classified, or regions cannot be classified into alpha helices or beta sheets. They might also be referred to as random coils. But these 2 are the main classifications for secondary structure. When it comes to tertiary, that's when we have a 3D unique shape. Now, a protein technically is active enough to function at a tertiary level, but the more the merrier, which is why when 2 or 3 of them are actually joined, that's when its efficiency is much better. And when 2 or 3 of them are joined, that's what we call the quaternary structure. So moving on to how we actually go from that DNA that we just spoke about to a protein, we have transcription and translation. So when it comes to transcription, we are going from DNA to a version of DNA that can be read by the following machinery, and the following machinery will then translate it into this polypeptide chain. So with transcription and translation, the topics themselves can be pretty, like overwhelming to start off with, but trying to think of them as three basic steps. So both transcription and translation, you have to, number one, start the process, number two, continue it, and number three, conclude the process. Have the basic structure, and then in your mind, just trying to map out what goes into each of those three levels. I hope it would make it a little less overwhelming to start off with. Eventually, there will be things that you might get confused with, because it's initiation and transcription, and then there's also in translation. But having those three will help you make sure that you're not forgetting any of the needed details. So getting back onto track, transcription, the first step, like I said, is initiation. The main thing that you need to initiate is the RNA polymerase, which will bind to a region upstream of template. So template is the region that you want to transcribe. Start unwinding the DNA and expose the template strand. Moving on to elongation, that's when it runs across, making an RNA complementary to the template strand. And if you notice here, there's the coding strand, then there's the template and the RNA. So your RNA is complementary to template, so is the coding strand, which means the RNA and the coding strand are exactly same, except that the T has been replaced with uracil. So understanding this would help you save time in your exam questions, because at times you might be trying to find complementary pairs for template if you're trying to figure out what the RNA is, but essentially the RNA is the coding strand, just that the T has been replaced with a U because it's RNA. Moving on to termination. So this is when it transcribes the terminating signal. That's when the machinery sort of dismantles and you end up with the DNA winding back to original, zipping up back to its original state, and a pre-mRNA. Notice how so far I've been calling it mRNA, but now I'm calling it pre-mRNA. For the sake of simplicity, we call it mRNA, but essentially it should be called pre-mRNA because there's some other modifications that happen to it before it can go else. And when I say go elsewhere, as in move on to the synthesis, the protein synthesis stage. But in terms of the post-transcription modification, there's three main things that are happening. The introns go into the bin, they're spliced out. The exons are excellent, so they are kept. The 5' end gets a methylated cap added to it, and the poly-A tail has a poly-A tail gets added to the 3' end. So this whole process is just to sort of give it a GPS to get out of the nucleus into the cytoplasm, and also to give it shield so things don't start attacking and degrading it. That's why we are bracketing both ends. Once we have done so, we'll move on to how this then translates into the amino acid chains, but it's important to note, when it comes to the amino acid chains, different amino acids can be synthesized via particular codons, but also different codons might be synthesizing for the same amino acids, which is why we have things like silent mutations, which we'll cover in tomorrow's lecture. But this is just a reminder about the degenerative code. Moving on to translation. Again, there will be three steps. The first step is initiation. So initiation is when the start codon is read in and you have the amino acid met, starting the process. Then we have elongation. So the machinery will keep running across. tRNA will keep bringing in amino acids based on the codon and anticodon sequences and keep getting attached. Eventually, a stop codon, one of these three, will be reached. And again, the machinery dismantles and you end up with a beautiful polypeptide chain. After we have the polypeptide chain, the other question might be, when does this process actually happen? So we have things tightly regulated to decide which genes turns on when, i.e. which gene will be translated and transcribed when. And this is actually really important if you think, because if we have all genes transcribed at all times, that's a waste of energy and resources. Sorry. And also you might end up with too much of substrate, too much of product at the end where you don't need that many, which again feeds into the fact that it would be a wastage of energy. So to make sure that we can make most of our resources, we can control which genes will be expressed and when. And in doing so, we recruit these regulatory genes. So regulatory genes produce proteins that can then control which genes are turned on and off. And in terms of genes being turned on and off, common example of those would be structural genes, such as enzymes, channel proteins, etc. So moving on to how we can actually control we have this region called promoter region in any gene. Then there's also exons and introns, which will, like I said, introns will not be translated. Exons are the ones that will be translated. This promoter region is where the whole process begins for transcription. But if we talk about how things are regulated in prokaryotes, we have this example of lac operon, which you guys might be familiar with. Important to note, sorry, I think I saw someone raising a hand. Okay, should there be any questions, feel free to put it in the chat. We'll just continue from now. As I was saying, with lac operon, this is unique to prokaryotes, not eukaryotic cells. It's more important in prokaryotes also because if you think about it, we don't have strictly defined organelles in prokaryotes. It's all just one big blob and everything happening there simultaneously. But in terms of the actual lac operon, it's a group of genes that control lactose metabolism. This is what it will look like. It's important for you to understand each individual component. So this promoter region is where RNA polymerase has to bind. It would usually be hit by this repressor, which would be bind to the operator gene. And these are the genes that you want to translate. Upstream of the promoter, you also have this cap site, which is essentially the binding site for cap, and it usually promotes the polymerase binding. When we have lactose, the allolactose, which is a form that it can be present in, would sort of take the repressor from here and take it out. And that's when the RNA polymerase can synthesize. When we don't have lactose available, the repressor will continue to bind to the operator, and RNA polymerase sort of hits a roadblock and cannot translate these genetic regions. So that overall is how gene transcription and then translation is regulated in lac operon. Moving on to the next topic, which is about the enzymes. So the enzymes facilitate any reaction by providing it alternative reaction pathways, i.e. are catalyzing the reaction. It's important to note when you define what an enzyme does, enzymes don't work magic, for lack of a better term. So they can't make the reactions happen which weren't going to happen anyway. Enzymes can only speed up processes that were going to happen at a slower rate anyway, but they can't induce a brand new reaction. And in terms of how enzymes do so, there's two main models. So there's the lock and key and induced fit model. You guys might remember. So lock and key suggests that the substrate, which is the thing that feeds into the enzyme's active site, is a complementary in terms of its shape and would straight away bind to it. However, in real life, unfortunately, things are not that easy. So the substrate's fitting actually is induced by molding the active site slightly, which is why the induced fit model is the one that is more commonly accepted these days. So that's how enzymes will fit in the substrate. In terms of factors that can influence its efficiency, temperature and pH, we have an optimal range for both of these. So up until the optimal range, it would most likely increase. It would be at its peak efficiency within that optimal range, and after that, it can actually come down. So if this graph were with temperature and pH on this axis, on the x-axis, it would actually be coming down after midpoint because, like I said, after the optimal range, it can be degenerating the enzyme. In terms of substrate concentration and enzyme concentration, this is not the case. So after the particular range where it's at its peak, as you can see, it sort of flattens or plateaus as a formal term, because we don't have a decrease in its efficiency. It's just that there's only so much that can happen. Imagine there being a X number of chairs and you and a group of friends are, choosing who's going to sit where. There's only so many people, there's only so many chairs. Same thing. There's only so much substrate and there's only so many enzymes. So it would keep going up. So the chairs will keep filling in as you have more people walking in. But after a point, there might be no more people or there might not be any more chairs left. That does not mean we have other people getting up, which is why this does not really come down. It just stays there. The other thing that can influence their activity is coenzymes. So coenzymes are known to improve their efficiency just by providing better sort of fitting for the substrate. Common examples of this include NADH and ATP, which we will cover in the next topic. Now, factors that can hinder the enzyme efficiency are inhibitors. So they usually prevent efficient binding of the substrate to the enzyme. We have competitive inhibitors, which will bind at the active site. We also have non-competitive inhibitors that bind somewhere else, but can still ruin the active site, which means the substrate will no longer be able to bind. So now we move on to photosynthesis. So just a breakdown of the topic. Photo light is used to synthesize energy at the end. Synthesize energy might not be the best term. It's synthesizing glucose really, because energy was there always. We're just converting it from one form to another. And in this process, like I said, we'll be synthesizing glucose, but obviously releasing oxygen and water as well. The main site of photosynthesis is chloroplast, which looks a bit like this. But before we get into the details of structure of chloroplast, it's important to note that there's this endosymbiosis theory that you will need to be aware of. So this explains that chloroplast maybe was an independent organism before and now has been engulfed into plant cells. And we have a list of evidence that suggests why this might be the case, which again, you can read in your own time. But moving on to the topic at hand, photosynthesis, there's two main steps, light dependent and light independent. The second step, I'm not a fan of the term light independent because technically this depends on the light dependent, which is why I sometimes would refer to this as the dark stage. Go with whatever you are more comfortable with, but just important to note that the light independent is indirectly still dependent on light, because without this one, this won't happen. But in terms of the details of each one, the light dependent occurs in the thylakoid discs or grana, grana, singular plural, and it starts with the chlorophyll capturing sunlight, converting chemical energy, and splitting water. So mainly hydrolysis is a main part of it. After we have hydrolysis occurring, there will be hydrogen and oxygen ions released, of course. And this is where oxygen would actually be released into the atmosphere as a byproduct. Then we move on to the light independent reaction. So this one, which again, like I said, I call the dark reaction, or some people refer to as the Kelvin cycle as well. So this would be the second stage of photosynthesis. It occurs in the stroma, and it, like I said, relies on the light dependent stage, which is why it technically does require sunlight. In terms of details that you need to know, if you are looking into more resources or your textbooks, surely there's much more information on these that you may be familiar with. For the exam, I would definitely recommend knowing the sites and at least the inputs and outputs of each stage. So in light independent, we had H2O mainly going in, which was then split up to give us hydrogen and oxygen. Oxygen was released and hydrogen got added to this NADP. The other thing we have released is obviously ATP, because like I said, during this hydrolysis, the main point of this was to produce ATP, which means obviously one of the inputs must be ADP and PI as well. We then move on to the light independent stage or the dark stage. This is where we have CO2, because one of the outputs is glucose. So we obviously need a source of C. So that's where carbon dioxide would be absorbed into the cycle. And we have hydrogen again. And the NADPH is just the carrier for hydrogen, which will be added on to this one, eventually coming off as NADP again. So again, try to have a table like this going on in your summary books as well. And try to justify where each input and output might be coming from. It'll help you have a holistic understanding of the topic. Then we move on to factors that can affect the rate of photosynthesis. So light intensity, carbon dioxide concentration, for both of them, it's the more the merrier. But obviously there's a limit to how much it can go up to, right? Because for both of these, there's a finite number of chloroplasts that a plant cell or plant overall will have. So there's a limit to how much carbon dioxide might be there, or how much the photosynthesis rate can increase up to. When we talk about temperature, again, there are enzymes involved which would help catalyze this reaction. So it would go up, it would hit this optimal range, but then at the end, when we have cooked up the enzymes, again, for the lack of a better term, the enzymes would have denatured, and that's when the rate, we actually see it decreasing. So let's apply all of that to this question. So essentially, this would be the question stem. Here are the graphs that this is referring to, and here are the 4 options. Because this is slightly more complicated than the previous one, we'll give you guys, let's have a look at the time. Yeah, we'll give you guys like 3-ish minutes. And as I mentioned previously, once you have an answer, feel free to chuck it into the chat.
I think you've stopped sharing, so we can't see the question anymore.
Sorry about that. There we go.
Awesome, thank you.
Alrighty, so we do have an answer in the chat, and the answer was D, which is actually the right answer. So just to walk through why it was D. So just looking at the graphs themselves, if you remember the axes, this would be the variable that we are changing, of course, and this is your dependent variable, which is the rate of reaction. But this graph resembles the graphs that we looked at for temperature. Or it could also be the graphs for pH, where we have the optimal range and the peak efficiency, and after that it actually decreases. So just based on that, option A and these two were, oh, this was temperature, but option A, which was Marcus, and option C could not be the answers because these did not have the right combinations in that this curve wasn't always referred to as temperature or pH, which is why I straight up went to option D. But in terms of the other options as well, if you look at these two, like I said, when it's about having enough mates to sit on X number of chairs or having X number of chairs to accommodate for all your mates, there is a peak level which you reach and then it sort of flattens. There's only so many people that can sit on so many chairs. So that's why these two graphs, you can't tell which one's enzyme and which one's substrate, but it could be either or. So technically speaking, even if option D had variable 3 as substrate and 4 as enzyme, that couldn't be the issue. But based on this, we can be sure that these has to be either or. In terms of why rather graph 1 was temperature, because when we increase the temperature, depending on the organism that the enzyme belongs to or its optimal range, the denaturing can actually be pretty steep, which is why this was more likely instead of variable 2 being temperature and 1 being the pH, where the degeneration might not be this steep. So that's why D was the answer. But in this option in particular, like I said, this combination of substrate enzyme for variable 3 and 4 was only in option D. So if I were to skim through these options, I would have straight away put D. If anyone has a better approach for this question, feel free to put it in the chat or happy to come back to it later on. But for now, we'll just keep going. So moving on to the next topic, which is cellular respiration. Okay, so the reaction for cellular respiration, one of the main things we need to know, and also gives away the purpose and what's actually happening as well. So the purpose is to break down glucose and produce ATP. In doing so, there's two main processes. There's aerobic respiration and anaerobic. In aerobic, we need oxygen. Anaerobic, we don't have enough oxygen. That's why. that's why the outcomes are also different. Moreover, the outcomes will be different for animals and plants as well, which we'll come back to later on. But important to remember that respiration occurs in 3 stages. There's glycolysis, Krebs, and etc. Moving on to the nitty-gritty details of each stage. Like I said, for photosynthesis, it would be important to understand where each stage is happening, what the inputs are, and what the outputs are as well. And then once you can justify them, I would say that is a decent enough understanding you need to tackle the exam questions. So in terms of the input for the first stage, which is glycolysis, you're breaking down glucose. So you would need glucose, obviously, for glycolysis. And glycolysis breaks down glucose into pyruvate, also giving you 2 ATP, and there's 2 NADH. These H's are eventually there to feed into electron transport chain, which we'll get to later on. But before that, we have Krebs cycle, which occurs in the mitochondrial matrix. The 2 pyruvates will then be taken in to produce CO2. We still have 2 ATP produced, fat and NAD molecules, which are all feeding into this chain. Here we now have oxygen involved, the NAD and fat molecules giving away the hydrogens. All of these feed in to activate, or rather help, the enzyme ATP synthase function. And once ATP synthase is functioning, as the name suggests, it would help you synthesize ATP, which is why the number of ATP we have produced is much more than the other 2. So that's overall the 3 stages of respiration. In terms of anaerobic respiration, things look slightly different for animals and plants, like I said. So plants, fermentation, common example we are aware of, obviously produces ethanol. But in animals, that's not the case. It's lactic acid that is produced after we break down pyruvate. And this is usually the case when the body is in a flight or fight mechanism, or when you are running on a treadmill, for example, or doing any other cardio sort of exercise. Because we have an increased demand of glucose, we just do glycolysis to send off the energy to different organs. The body does not have time to efficiently wait for a lot of oxygen to be available and then do the respiration, which is why it would quickly just try to generate some ATP. And in doing so, one of the outputs would be lactic acid, which again partially contributes to how we feel afterwards. And here's a table just breaking those down for you and a bit more neatly compared to the texts. And this is just a reminder of the mitochondria. So mitochondria, again, the theory of endosymbiosis has been applied on mitochondria as well, for the similar reasons that it has its own circular DNA, there's ribosomes present as well, and the structure as well, how its compartmentalization sort of resembles. And again, this diagram, something that you are familiar with, obviously, but just a reminder, this is what I referred to by the matrix, and this is what the sister membranes are. And yeah, other than that, I don't think we need to know much more on mitochondria. So we'll just finish cellular respiration with the factors that impact. Again, glucose and oxygen concentration, it flattens. And with temperature, again, because of denaturing, it actually comes down. So the last thing about area of study 1 for unit 3 is cellular signaling. So cell-to-cell communication, there's 3 main types: endocrine, paracrine, and autocrine. Again, the diagram does a pretty good job at explaining what the differences are. So endocrine is via bloodstream, paracrine is a neighboring cell, autocrine, auto means itself. In terms of some examples, so we have neurotransmitters, hormones, cytokines. Again, their functions have been listed over here as well. And so are the mode of communications. Exocrine, you might have noticed, is a new one because pheromones can be released from one organism and cause a response in another one. That's why exocrine, because it's exiting the organism. Other than that, all the other signaling molecules example that we see fit into the 3 categories of communication that we just covered. In terms of signal transduction, there's 3 main steps. 1, there's reception. 2nd is it has to be transduced, translated sort of. And 3rd is the actual signaling response. In terms of transduction, the main thing that's happening is amplification of signal, for which we need secondary molecules. But before we even get into that, oops, sorry, a little too early to get there. Yeah, so before we get into that, there's signal reception, which will depend on the nature of the molecule. If it's a hydrophilic molecule, i.e. a water-loving molecule, its receptor will have to be outside, because remember how we went through for phospholipid, there's hydrophobic regions sandwiched between the hydrophilic heads, and they will not be very nice to this hydrophilic molecule. So that's why the receptor will bound to the outside cell membrane, and then secondary messengers will take care of things. If it's hydrophobic, it can actually pass through and bind to intracellular receptors. Regardless of the two types, the second stage would be amplifying the signal, and third would be the actual signaling response that you want from the cell. This whole cell-cell communication has been sort of elaborated further using this 5-step model, in which we have a particular stimulus, then we have a certain receptor, then the signal is transduced, then we have effector, and this, apologies for the typo, should actually have step 5, because that's actually the response that we have generated. An example of when cell-cell communication might be really important is apoptosis, cell death, or cellular suicide, as they say. There's multiple triggers for apoptosis, such as these ones. And because of finger webbing, we see how apoptosis is not always a bad thing. Sometimes it's needed as well. And this is a classic example of why we need efficient communication, because if when a cell should be killed is not killed, it can lead to further consequences. Unfortunately, things like cancer as well. But in terms of apoptosis, again, we have two pathways. There's mitochondrial, which is where the signal will come from inside of the cell, or there's the extrinsic pathway, which is when there will be a ligand binding to the death receptor. In either case, you have a caspase set activated. So if it's mitochondrial, there will be cytochrome C release, which will then activate caspases. So caspase, anything that ends with an ase usually is an enzyme. So caspases are the enzymes. They would then feed into a signaling cascade, which is the process, and eventually called shell shrinking and blebbing. In this one, we don't have cytochrome C release, we have other signaling molecules, but essentially activating the caspases, introducing a signaling cascade, which has a response causing cell shrinkage and blebbing. Okay, before we move on to antigens and pathogens, let's take a break for a few minutes. So it's 46 right now. We should be able to resume at 6:55 for the next part. Meanwhile, I'll just have a look in the chat if we have any questions. If not, feel free to just go grab yourself a drink or something, and then we can get started with area of study 2 in 10-ish minutes. Alrighty, so welcome again, everyone. So we are moving on to the second area of study now, and I would like to introduce you all to Eve, who will be taking it on from here.
Okay, so hi everyone, my name's Eve and I'll be going through the immunology section. So at first, I just wanted to revise a little bit about antigens and pathogens. So for antigens, if you look at the study design, it defines it as a unique molecule or part of a molecule that is able to initiate an immune response. So basically, just simply speaking, it's just anything that can initiate an immune response. And we can divide antigens into self-antigens and non-self-antigens. So for self-antigens, these are molecules or parts of molecules that are found on our own cells. And in normal circumstances, the immune system will hopefully recognize them as self and not mount an immune response against these self-antigens. For non-self-antigens, these are the foreign molecules. So these can be things such as like pathogens. So this includes like viruses and bacteria. These are hopefully recognized by the immune system as non-self, and an immune response will be activated to try and eliminate the non-self antigen. An example of a non-self antigen is actually an allergen. So an allergen is a normally harmless antigen, but in people who are allergic to something, they'll produce this abnormally strong immune response. So examples of allergens are things such as like pollen or nuts, or if you have like hay fever, it's coming to like spring right now, pollen is an example of an allergen. So now let's talk a bit about pathogens. So pathogens are living or non-living agents that cause disease. So they're an example of a non-self antigen. And for some non-cellular pathogens, this is just a table that kind of goes through a little bit about some different non-cellular pathogens. So this includes viruses, viroids, and prions. And I might just explain a bit about viruses. So viruses, in terms of their structure, they have this protein coat or capsid that encloses the nucleic acid. So if we remember, the nucleic acid is the genetic information. So it's the thing that's encoding the proteins. and it can be DNA or RNA. And then sometimes around the capsid, you have this lipid envelope. So examples of viruses are things such as like HIV, influenza, or even coronavirus. So with viruses, it's really important to remember that they aren't able to replicate on their own. So they require a host cell. That's because they don't contain the machinery, for instance, like ribosomes to make proteins. So in order for a virus to replicate, they actually have to infect a host cell. Now we'll move on to cellular pathogens. So these include things such as like bacteria, protozoa, fungi, and parasites. So with bacteria, they're prokaryotes, so they're unicellular, and they usually, for their structure, they have like a cell wall, then a cell membrane, and inside they will have a single circular chromosome, as well as the ribosomes and plasmids. So bacteria, unlike viruses, they're able to replicate by themselves, and they don't actually have to infect a host cell all the time. Examples of bacteria are things such as like tetanus, typhoid, also things like salmonella or meningococcal disease. And bacteria, they replicate by binary fission. So that means that one bacterial cell will actually divide into two genetically identical bacterial cells. And for our bacteria, we can actually treat them by using antibiotics. So for antibiotics, you can use them to treat bacteria, but you can't use them to treat viruses. So moving on, we'll talk about innate immunity and adaptive immunity. So it's really important to distinguish the two. So for innate immunity, this is the immune response that will occur first. It's non-specific, meaning that if you encounter a pathogen, the same response is going to occur to every single pathogen. It doesn't really distinguish between different pathogens. And also no memory is formed, which means that if you are exposed to, let's say, the same pathogen again, the innate immune system isn't going to remember that that's happened, and it's just going to produce the same response. Okay, so for the innate immune response, it encompasses like the first line of defense and the second line of defense. The first line of defense is kind of, I like to think about it like it's trying to prevent the pathogen from entering the body in the first place. So that's things like your skin. It's also things like the mucociliary escalator. So if we're thinking about the respiratory system, we know that there's like mucus lining our airways, and the cilia are like these little hair-like projections that are on the cells of our airways. And they kind of beat in this fashion to sweep, in a way, the mucus up out of our airways. And we end up like coughing that out or swallowing it. So if you think about when you have like a cold and that phlegm you produce, that's because of the mucociliary escalator. And that helps to kind of like remove pathogens from your airways. Then you also have things like, so chemical barriers such as lysozyme, defenses, as well as the low pH in your stomach. The host microbiota, so this is the organisms in your gut, they can also act as a barrier because they will compete for like things like nutrients or space with the pathogens that might try to invade your gut. For plants, they have things such as the waxy cuticle of leaves and bark, and they also have things such as enzymes and oils that also help to try to prevent the pathogen from invading them. So for the second line of defense, we're thinking about the different cells. So these are the white blood cells and also complement proteins. And importantly, because it's part of the innate immune response, we have to remember that these defenses are non-specific. So if you wanted to talk about some cells, first we have some macrophages. So macrophages are found in the tissue, and they are professional antigen presenting cells. So this means that when they come across a non-self antigen in the tissue, they're going to take up the antigen. So by phagocytosis, so this is what we covered earlier. This is the process by which the macrophage is going to use its membrane to engulf the pathogen and take it up. And then they're going to present the non-self antigens on their cell membrane. And they use that using a molecule, which is called the MHC class 2 molecule. The macrophage can then go on to present those antigens to lymphocytes, but they can also just produce inflammatory mediators such as cytokines. For neutrophils, their primary role is phagocytosis, and they're usually the first cells at the site of infection. And if you think about sometimes when you get a cut and you know that sort of like pussy yellow substance you get, that's actually the dead neutrophils and also like the cellular debris and the pathogens that is kind of phagocytosed. That's what the pus is made of. So for mast cells, they're involved in allergic reactions and kind of the inflammatory response. So mast cells inside their cytoplasm, they contain these granules, and these granules contains molecules such as histamine. So histamine is kind of the reason for why you see those different symptoms, such as like redness, warmth, swelling, when you get like an allergic reaction. So they will cause things such as vasodilation. So vasodilation means that your blood vessels become, they expand. So this means that more blood will go to a certain tissue area. And as a result of that, you will get the redness. So if you know, think about sometimes when you cut your arm or something and it becomes really red, that's because all the blood is rushing there. And that blood is bringing all the different immune cells and immune mediators to the site of infection. You also get increased vascular permeability. So that just means that normally your blood vessels, they have like holes in the blood vessel, and they don't usually allow too many things to move from the blood vessel into the outside tissue. But with histamines, they will increase kind of the size of the holes in the blood vessel, meaning that more things can move from the blood vessel into the tissue. And for the immune response, this really helps because it can help the different immune cells, so the neutrophils, move from the blood vessel into the tissue. But at the same time, it will also cause a lot of fluid, which we call interstitial fluid, to move into the tissue, and that can cause swelling. So if you think about your cut, sometimes you will notice that it's quite bumpy. That's because of the fluid moving into your tissues. So for dendritic cells, these are also antigen presenting cells. So they're also involved in engulfing the antigen by phagocytosis, then migrating to the lymph nodes and presenting it to the different lymphocytes in the lymph nodes, and kind of activating the T cells and B cells. So importantly, dendritic cells, they're a really important link between innate and adaptive immunity. So they're kind of the bridge, if that's the way you want to think about it. So now we're talking about complement proteins. So once again, complement proteins are non-specific because they're part of the innate immune system. So complement proteins, they're made in the liver and they're activated during inflammation and they circulate in the bloodstream. And they have multiple functions. One function is opsonization. So this is where they kind of act as like a flag. So they bind onto antigens on the pathogen. They kind of act as a flag that like waves and signals to other immune cells that this is like a pathogen and you can come and like eat this pathogen. There's also things such as the membrane attack complex. So this is when it's going to poke like holes, for instance, in the bacteria's membrane, and that's going to cause the bacteria to like lyse and die. Another function is also agglutination. So this is when you have antibodies that will kind of clump pathogens together, and that also makes it easier for other immune cells to come and phagocytose the bacteria. So I mentioned inflammation before. This slide's just kind of outlining the steps in a more clear way. So first you'll get the tissue damage, then you'll get the mast cells that release the histamine that triggers the inflammation. Then you're going to get the vasodilation, which causes the redness and the warmth, and then the increased vascular permeability. So that's the fluid leaving the circulation to enter the tissue. And this is going to cause the swelling that we see. With this, you can also get pain during inflammation. And that's because you get like nerve endings in your skin that are like sensitized, and that causes pain. So now we're going to talk about the lymphatic system. So this is really important because if you think about inflammation, for instance, because of that increased vascular permeability, you're getting so much fluid that's just leaking out of the circulation into the tissue. And you need some way to kind of return that back to the blood system. So that's what the lymphatic system does. It collects that fluid, which we call interstitial fluid, that leaks out of the circulation, and then it brings it back to the vascular system. And in the lymphatic system, there's these masses of lymph tissue called lymph nodes, and they're really important because they kind of act like these areas of surveillance that screen the fluid that's passing through the lymphatic system for any non-self antigens. And if they detect that there's any non-self antigens, they're going to mount an immune response. So these lymph nodes contain like lymphocytes, so the T cells and B cells. And if you've ever been sick and you've like noticed like sometimes in your neck you feel like these lumps, that's actually the lymph nodes in your neck becoming enlarged because they're like being activated and all the T cells and B cells and lymph node are like proliferating, which makes the lymph nodes like swell, so you can feel them. So now we'll move on to adaptive immunity. So this is the one that is specific, which means that it will mount a response that is specific towards a specific antigen. And it also is the one that forms memory, which means that if you are exposed to the same antigen again, an immune response is going to be mounted that is greater and faster. So yep, this is just what I mentioned before. So with the adaptive immune response, it's mainly mediated by the B cells and the T cells. So the B cells are the cells that mature in the bone marrow, and there's two main types. There's B plasma cells, so these are the ones that produce the antibodies, and there's also the memory B cells. So these are the ones that keep the memory about the antigen. For the T cells, they mature in the thymus, and there's three different types. There's the T helper cells. So these are the ones that kind of provide help to other white blood cells. There's also the cytotoxic T cells. So these are the ones that are, for instance, involved in like viral infections, and they're going to release enzymes that lyse cells. Finally, there's the memory T cells. So these are the ones that remain in the circulation, and they also maintain memory of the antigen. So talking about antibodies, so these are the proteins produced by the B cells. They are specific proteins produced by B cells in response to specific antigens to which they bind and inactivate. So the key word here is really specific. It's really important to remember that one B cell is specific for a specific antigen and is only going to produce antibodies that are specific for that antigen. And examples of antibodies are there's like many different types. So IgG is the main type of antibody that's circulating in the blood, but there's also IgE. So IgE is the antibody that's found in allergic reactions, and we'll talk a bit about that later on. So you can kind of classify adaptive immunity into two main branches. So there's humoral immunity. Humoral immunity is mediated by B cells, and it involves the function of antibodies. The other branch is cell-mediated immunity, and that's mediated more by T cells. So for humoral immunity, this flowchart kind of just gives the steps of what happens. So first, you're going to get your invasion with a pathogen. Then your dendritic cells that are in your tissues, they're going to detect that pathogen as non-self. They're going to engulf the pathogen and then display the non-self pathogen antigens on their cell surface. Then they're going to move to the lymph nodes. And then in the lymph nodes, these dendritic cells are going to present the antigen to the B cells with the specific B cell receptor. So this B cell receptor is specific for this antigen. Through antigen presentation, the B cell then becomes activated and it will differentiate and then also migrate to the site of infection. So the B cell can differentiate into either the B plasma cell. So these are the ones that produce the specific antibody, or they can differentiate into memory B cells. So these are the ones that will retain memory about the antigen. So for cell-mediated immunity, it begins with the same sort of steps initially. But here we're getting the dendritic cells that have migrated to the lymph nodes. They're now activating the T cells with the specific T cell receptor. So the T cell is going to undergo activation, differentiation, and is also going to migrate to the site of infection. So the T cell can differentiate into memory T cells. So these are the ones that retain memory about the antigen. They can also differentiate into T helper cells. So T helper cells, as the name suggests, it provides help to other white blood cells. So by providing help, it kind of increases the efficiency of the response that white blood cell is able to provide. With cytotoxic T cells, they're the ones that release lyse infected cells by releasing enzymes. So this table just kind of summarizes the key differences between the innate and adaptive immune response. Okay. So now we're going to talk about some applications. So first we're going to talk about what's the difference between passive versus active immunity. So passive immunity is when the individual doesn't actually mount their own immune response, but they will receive antibodies from another external source. So because the individual isn't actually mounting their own immune response, they won't form any antibodies, and they're not going to form any memory either. In contrast to this, with active immunity, the individual is actually going to produce their own immune response. So they're going to produce their own antibodies, and also they're going to produce memory B cells. So this means that they're going to be able to remember that they've been exposed to a certain pathogen. And if that pathogen reinfects the person, they're going to be able to mount a greater, faster response. And then comparing natural and artificial immunity, natural just means that it's through natural exposure to the pathogen, whilst artificial just means it's through artificial means. And here's some examples. So if we remember, passive immunity means that you're not forming any memory and you're not producing your own antibodies. So an example of this is for natural passive immunity. This is like when you're a baby, you drink breast milk from your mum, and in the breast milk, there's like antibodies. So these antibodies will pass into the baby, and they will be able to protect the baby in the case that the baby becomes exposed to certain pathogens. And that's because the baby, well, when you're a baby, you haven't developed an immune response. Like your immune response hasn't matured yet, so you aren't be able to make your own antibodies. In terms of a artificial passive type of immunity, that's for instance when you get, for instance, a snake bite. The doctor might give you an injection of anti-venom, so that's like a type of antitoxin. And the anti-venom will contain antibodies that have actually been produced in another animal. So it's usually like, I think, horse or rabbits. And that antibody will be able to neutralize the venom that is in your body. So for active immunity, so this is the one where you have been, where you mount an immune response yourself towards the pathogen and you do form memory. For natural active immunity, this is just natural exposure to the pathogenic antigen. So it's kind of what we've been talking about so far. For artificial active immunity, an example of this is vaccination, and I'll talk about that more now. So vaccination is kind of when, if you think about, for instance, the COVID vaccine that everyone's talking about right now, the vaccine will contain the non-self antigens of the COVID virus. So it's probably just certain parts of the virus in the vaccine. And what happens is when you get the COVID injection, the body is going to recognize those non-self COVID virus antigens as non-self, and then it's going to mount an immune response against that. And the important thing about vaccination is that it can help to achieve herd immunity. So this is kind of the concept where a sufficient proportion of the population is immunized so that we're able to protect people who can't be immunized against a certain disease. So this is kind of why people have been talking about with COVID, trying to reach that like 70, 80% margin of people being vaccinated, because we're trying to achieve like herd immunity and protect those people who can't get vaccinated. And then this graph here is kind of just showing that, so the primary immune response here, this could be you getting your vaccination. And at this point, the antibodies that you can see here that the response is not as big and it takes longer to occur. But then if you did get infected yourself post-vaccination, the response that you produce will actually be greater and faster. And this is kind of how vaccinations will help prevent us from getting serious like disease. So now just talking about autoimmune disease. This kind of happens when your immune system isn't doing what it should do. So like I mentioned before, usually with self-antigens, we want to recognize self-antigens in self. We don't want to attack self-antigens because that means we're mounting our immune response against ourselves. But sometimes this does occur. And when that occurs, we call that autoimmune disease. An example of this is in multiple sclerosis. So that's when your immune cell system specifically T cells, they destroy the myelin sheath of nerve cells. So myelin sheath is kind of this substance that surrounds the axons of your neurons, and it just kind of helps in conducting nerve signals down the neurons. So it's really important for like simple movements even. So because the T cells are destroying that myelin sheath, that means that your nerve signal cannot be conducted effectively down the neuron. And as a result, you're going to get things such as like vision loss, pain, impaired coordination. So a lot of your neurological functioning is going to be affected. For immune deficiency, this just means that you have a reduced responsiveness of your immune system. So an example of this is HIV. So HIV is a virus that actually infects T helper cells. So the target host cell that infects is T helper cells. So as a result, because T helper cells are so important as part of the adaptive immune response, because they're no longer able to function and they're being like killed off by the HIV, this means that the adaptive immune response isn't able to work as efficiently as it could. And as a result, you get reduced responsiveness of your immune system. So HIV is transmitted through bodily fluids such as blood, and it can lead to AIDS. So we mentioned the allergic reaction before, but I was just going to talk a bit about it. So like I mentioned before, it's kind of a response to a normally harmless antigen, and it causes this abnormal immune response. So with allergic reaction, you get two stages. So if we're talking about, for instance, pollen, during the first stage, this is sensitization. So this involves the first exposure of the person to pollen. So what happens is that the pollen, like any antigen, it's recognized as foreign by the body, and then this leads to all those series of events that we talked about before, which leads to B and T cell activation. The T cells are going to provide help to the B cells. And then this is going to allow the B cells to produce IgE antibodies against the pollen allergen. And what happens with these IgE antibodies is that they actually become embedded in the membrane of mast cells. So you can see here, the mast cell, the IgE antibodies have become embedded in the cell membrane of the mast cell. And what happens is that when you are exposed, so this is the second exposure, when you are exposed to the same pollen allergen again, this pollen allergen is now going to bind, so you see here, it's going to bind to the IgE antibodies on the mast cell. And what it's going to do is that it's going to cross-link these IgE antibodies. That's going to cause the mast cell to be activated, and the mast cell is going to degranulate and release all that histamine. So like we mentioned before, histamine causes all that. It causes vasodilation, it causes increased vascular permeability. So that's going to cause all those allergy symptoms that we can think of. So that's going to cause the itchiness, the redness, the swelling that we notice when people have allergies. So monoclonal antibodies, these are antibodies that are produced by a single clone of B cells. So the monoclonal just means that it's produced by a single clone of B cells. And as we would expect, because they are produced by a single clone of B cells, they're going to have a single specificity. So they're only going to recognize one specific type of antigen. And what's really useful about monoclonal antibodies is that, for instance, in cancer, you can attach actually a drug or whatever you like to the monoclonal antibody. And if the antibody is specific for the cancer cell, that drug or toxin is going to be delivered directly to that cancer cell. So that drug or toxin, it can do things such as bind receptors on the cancer cells, and this can lead to it can inhibit the growth of the cancer cells, so prevent it from proliferating, or you can do things such as induce apoptosis, so kill the cancer cell. And what's really cool about this is that because the monoclonal antibody is specific only for the cancer cell, it's not going to attack your own cells. And that's really important because we want to make sure that we're removing the cancer cell, but we don't want to also attack our own cells in the same place. So this is just a quick question. And I'll give you guys maybe a minute just to complete that. And please put your answers in the chat. if you have an answer for this. Okay, so it looks like we have two answers. So B. Yep, you guys are correct. So dendritic cells, yes, they are antigen presenting cells. So they're going to present those antigens. They're going to migrate from the tissue to the lymph nodes and then present the antigens to the T helper cells. So if we look at A, macrophage stimulate inflammation by secreting interferon. They don't usually secrete interferon. That's more of a product of T cells. For mast cells, engulf bacteria and debris. They don't actually undergo phagocytosis. That's more of what macrophages and neutrophils and dendritic cells do. So mast cells are the ones that will release those mediators such as histamine in like allergic responses. And for neutrophils, they don't secrete antibodies. That's what the B cells do. So neutrophils are actually the ones, like we mentioned before, they're the ones that undergo phagocytosis. So yeah, good job, everyone. Yep. So that's actually the end of our revision lecture. But if anyone had any questions, please feel free to ask in the chat, or you can unmute if you like and ask directly. That's okay too. Otherwise, I might just mention that we do have our Unit 4 lecture tomorrow. So if you guys wanted to come along to that, and we can talk to you guys about the Unit 4 content and revise it together. Also, you can sign up for other subjects revision lectures that TFI is also providing, if you guys would like to. Did anyone have any questions at all?